Hello everyone, my name is Tui Wen. Today I would like to speak to you regarding the medicinal use of plant antimicrobial agents in humans. We all are well aware of the antibiotic therapy that we've been using to treat all kinds of infections, from the over-the-counter topicals such as bacitracin ointment, neosporin ointment, Bactroban, to the oral antibiotics, and then the intravenous antibiotics. Today we are going to look at a different modality, not a new modality, but a different one to help us to address the issues of infections. We are going to look into biosynthetic plant molecules instead of the synthetic antibiotics that we are so used to. But first, let's go over the basic plant biology so that we could understand where these molecules come from. The photosynthesis that actually produce a lot of essential primary metabolites for the growth and development of plants, such as glucose, fatty acids, amino acids. Photosynthesis is mostly occurs in the leaves of the plants. What we are interested in today are the plant volatile oils that many of us are so used to go into the garden and smell the roses. These volatile oils are not just for a good smell or aroma. They are found mostly in the flowers, but they could also be found in leaves, bark, fruit, seeds, stems, and roots. These are called secondary metabolites. They are not at all involved in the growth and development of plants, but they must be there for a purpose, because in order to produce the volatile oils, the plants have to use energy to form these chemicals, and therefore they do not want to waste energy for nothing. So first, in the photosynthesis, the plants form molecules through different cycles, such as the Calvin cycle, the Krebs cycle, the acetate malonate pathways to form fatty acids. The two pathways that we are interested in with the volatile oils are the phenylpropanoic pathway, whereby the aromatic compounds are formed, and the word aroma means that the chemical contains a benzene ring. Another pathway is the acetate mevalonate pathway. The terpenoids and the steroids are formed in this pathway. So to make it simple, the volatile oils are categorized into two families. One is the terpenes and the terpenoids, and the second one is the phenylpropanoids. The terpenes are 5-carbon atom molecules. They can build upon that, so we can have isoprenoids, that our molecules are built upon this 5-carbon unit. We can have monoterpenes with 10 carbons. We can have sesquiterpenes with 15 carbons. The terpenoids are oxygenated derivatives of terpenes, and we can find them in lavender, sandalwood, geranium, clary sage, and eucalyptus. In the phenylpropanoids, we can have them in the group of phenols, like clove bud, aromatic alcohols like phenylethanol in rose absolute, aromatic aldehydes as in cinnamon leaf, or ethers as in sweet fennel oil. So the volatile oils are considered to be a very small amount of chemicals that are produced by only about 1% of the plants. And the production of volatile oils by plants is an energy-requiring process. So why do plants make volatile oils? For what reason? So plants use volatile oils for possibly to defend themselves from the herbivores, or to actually help them with pollination from the bees and the insects to enhance their reproduction. They also use volatile oils to compete with the nearby plants for light, nutrients, and space. They also can use it as a messenger to send out signals to attract the parasites to come in and rescue them when they are under attack. And sometimes this is called an SOS signal. They also can use volatile oils to repel the insects. And the example for this is wintergreen oil and citrus oil. What we are interested in today is the use of the volatile oils to prevent the growth of plants pathogens such as bacteria, fungi, and viruses. So plants use these secondary metabolites for all of the above purposes. One is called the phytoanticipants. These are the chemical molecules that were preformed by plants to help them as a barrier for microbial attack. The second one is called phytoalexins. They are induced when the plants are attacked by the microorganisms. So for the phytoanticipants, a couple of examples that they were used by plants for an antifungal effect. The evanescens as a oak root saponins, and in tomato is the alpha-tomacin. For phytoalexins, the examples here are the hydroxycoumarin, scopoletin in tobacco plants that have antimicrobial properties. Also some other chemicals in rice, in the crucifers, and in the other plant species that help to abolish bacteria, viruses, and fungi. So why is the concern of trying to find another approach in taking care of infection is because it is now becoming public health issues. A lot of microorganisms now become multi-drug resistant, and particularly the gram-negative organisms. And nowadays, MRSA is so prevalent in the hospital environment, and so many thousands and millions of dollars have spent in taking care of these organisms and infections. So how do these phytoanticipants and phytoalexins help in the treatment of infection? They are thought to be served as antibiotic potentiators. That means that they assist the current antibiotics that we use to gain back some of the therapeutic effects that were destroyed by the microorganisms. They could also be served as virulence attenuators. In this case, they assist the host immune systems to respond to the pathogen invasion. And thirdly, this is not due to the toxicity or the chemical reaction of the phytoalexins or anticipants, but it's due to the fact that they prevent the bacteria or microorganisms to adhere to the human tissue. So in the antibiotic potentiators, they could serve as inhibitors in, for example, the gram-positive bacterial efflux pump to prevent them from being resistant to the antibiotics. An example in this case is the catechin found in green tea extracts that abolish the tetracycline resistance in staphylococcal isolates. Another activity is acting on the cell wall and the cell membrane of the bacteria, and thereby destabilizing them. Example in this case is the terpenoids works synergistically with the antibiotics to enhance the susceptibility of the MRSA toward beta-lactams by interfering with the bacterial cell wall peptidoglycan biosynthesis. Serving as virulence attenuators, they can block the bacteria from adapting to the mammalian environment to become resistant to that. They can also prevent the microorganisms to produce toxins to the host. An interesting concept is called quorum sensing system. This is a system whereby the more the bacteria multiply, they produce toxins and hydrolytic enzymes that they prevent the host to have an early detection of the invader, and thereby the host would not have time to build up the immune system. This virulence attenuators, the quorum sensing system, is common in the biofilm formation or the colonization of the wounds that we see very common nowadays in the wound care setting. An example for the inhibitors of this quorum sensing system is the garlic extract. The garlic extract was shown to reduce the Pseudomonas aeruginosa biofilm to the tobramycin treatment. So patients with chronic open wounds that come to the wound care center every week, they are debrided to remove the colonization or the bacterial biofilm. They have done studies that show that this process, the quorum sensing mediated process, needed for the colonization of the pathogens such as Pseudomonas in the respiratory infections of cystic fibrosis. The third concept sometimes could be missed in an experiment is called the disruption of adhesion. In this process, the phytochemicals prevent the adsorption or the adherence of the microorganisms to the host cells, thereby preventing the microorganisms to have an effect on the host. So, for so many years, we have used holistic medicine to treat a lot of diseases. There are lots of clinical applications, such as the use of Terminalia tubula, which has the gallic acid derivatives that would help to treat the infection of methicillin sensitive as well as methicillin resistant Staphylococcal aureus. An Ayurvedic herb, Sandravati, that helped to reduce the acne vulgaris lesions. Tea tree oil was used to reduce the skin acne. Extract of Solanum nigrescens were used as intravaginal suppositories to treat Candida albicans vaginitis. The neem is used also to treat Candida, and the arjuna is used for wound healing, and aloe vera was used to treat psoriasis. So, for centuries, different cultures have used plants to treat infections and other illnesses. However, controlled clinical studies are very scarce. What we need now is we need more control and randomized clinical trials of the use of these phytochemicals in humans. So we need to have a good collaboration of the traditional healers and the trained scientists. Thank you very much for your attention.